Hello, we are the University of Lethbridge Collegiate iGEM team, and we are excited to share our project with you. Club Squared, a means to remove the infectious root from club root. Our team consists of 20 undergraduate student members and 5 undergraduate advisors. We are lucky to have Dr. Vineet Rathod as our primary investigator. Additionally, we are very grateful for our funding agencies, the University of Lethbridge, Corteva, and Genome Alberta this year. So what is clubroot? Well, clubroot is a soil-borne protist pathogen that causes the development of clubs, or galls, of varying sizes on the roots of brassica crops. These galls result in nutrient deficiency in the plant and eventually result in plant death. The pathogenic spores produced by clubroot can remain dormant for several years, making the infection difficult to eliminate. Canola or Brassica napus is a common crop in Alberta that falls victim to clubroot infection. Considering that canola farming is so prevalent in our province, clubroot infections result in major yield losses. What's extremely concerning is that 20 years ago, clubroot had a prevalence of 0% in Alberta, and now 67% of our counties and districts are known to be home to clubroot, emphasizing the spreadability of the disease. Many researchers and agronomists are concerned with the abundance of clubroot in our province. Hence, Corteva, an agri-science company, approached us and tasked us with creating and researching a solution to this issue. Furthermore, many of our team members have grown up in Alberta, and many are related to farmers who are directly impacted by the issue we are attempting to solve, making clubroot an area of interest academically, but also personally. The optimal conditions for clubroot to thrive are acidic soils with a pH of less than 6.5, making Alberta a place for the pathogen to prosper and proliferate. Additionally, wet conditions favor clubroot, which also has the ability to survive in cool and warm climates, making it flexible to different conditions. The spread of clubroot is often facilitated by the movement of machinery and equipment between different farming plots, winds, and water erosion, as well as the use of other infected crops as feed. There exist a few current methods to detect and mitigate the spread and subsequent infection of clubroot. These include crop rotation, equipment sterilization, and the creation of genetically resistant crops. However, these techniques are not always the most effective or sustainable considering the resilience of clubroot and its dormant spores. Furthermore, the current method used to test for the presence of infected clubroot is through DNA polymerase chain reaction, or PCR, which requires farmers to ship their soil samples to labs and wait for the results, which often takes weeks. Additionally, each PCR test costs $95, making it not very economically friendly. Therefore, the current testing methods are costly, laborious, and do not have a fast turnover with regards to the detection of pathogenic clubroot.
In order to get to the root of this pathogenic protein, we must understand this pathogen's life cycle. What makes clubroot such a destructive pathogen is its way of reproduction and how it can spread and affect nearby crops. It replicates via zoospores, which can remain dormant in soil conditions for decades. Once optimal soil conditions arise, the zoospores can start infecting crops like canola, hijacking the host's nutrient uptake for itself, which leads to the production and release of more zoospores, and resulting in the death of the host. To briefly describe what our team has been working on and showing our preliminary results, the main project for this year's U Lethbridge iGEM team is to create a direct detection kit which can detect clubroot in soil well before crops are planted. We want our solution to be simple and easy for farmers to easily test soil samples for clubroot while also being very cost efficient. Initially, one of our 2 goals was to create an ELISA test kit to be able to detect clubroot in soil samples. The biggest feedback received from our community, judges, our mentor, is that an ELISA test kit would be impractical for farmers to use in the field, and we require a proper lysis protocol, and possibly even sending samples to a lab. All these steps would reduce the efficiency and basically be homologous to sending samples out for a PCR test. We have expanded our test to be like a take-at-home rapid antigen test. Parallelly, our second hypothesis was to use our engineered chimeric protein that is specific to clubroot as a strategy to mitigate its spread and the formation of the galls and increase crop yield for farmers. After multiple interviews, our team decided to change this goal as a long-term target, as it is not feasible to be done for the time of the competition. One of the challenges is that the whole genome has not been sequenced. Secondly, the life cycle makes it very difficult to target dormant zoospores. A way for targeting the pathogen without harming the host would be rather difficult, and there are no such protein structures solved to see the structural homology of pathogenic clubroot in various crops. That is not to say we have given up on this goal, as our detection system has considerable overlap of what our target for mitigation would be, a protein called PBEL04. So we were hoping to create a successful direct detection kit this year, and using the data we get from this kit next year, we will move forward with mitigation.
Well, how was our project developed? Like stated in the previous slide, we have expanded our test to be like take-at-home rapid antigen test. We have potentially developed an engineered chimeric flow body that can directly detect with the clubroot infection in soil samples. Our target is a protein called PBEL04, which is highly expressed during infection of hosts and gall formation. Initially, we had 2 possible protein targets, and we have narrowed them down to PBEL04 as it is specific to clubroot and important in the life cycle of clubroot, making it a great potential target for future mitigation plans as well. Our engineered chimeric flow body will bind to PBEL04, the targeted antigen, through protein interactions, as you can see from the right of the screen. Our idea is to have test strips where we can see if our engineered chimeric flow body binds to PBEL04, giving the strips 2 bands. If our engineered chimeric flow body, which is linked to a detector molecule, binds to the antigen. To emphasize, this detection kit is our end goal. We must first ensure that our engineered chimeric flow body is interacting with PBEL04, and we will do that through testing through indirect ELISA tests. If our ELISA test shows successful protein interactions, we can move forward to creating our test strips for farmers to use.
Now, let us switch gears to our prototype, which are our constructs, engineered chimeric flow bodies, and preliminary results on the interaction of the flow body to the target protein. The team has designed three main constructs, where the first one demonstrates protein-protein interactions with PBEL04 by binding to one of its three common domains. This construct is tagged with GFP, and if the protein successfully binds to PBEL04, it will show double band on the test strip shown previously, confirming the presence of club root. The second construct is very similar in which it is basically the same construct, but instead of tagging the construct with GFP, it is fluorophoric tagged. This was done by adding a lysine-rich region where an alexafluor can attach. This change was logical to the wet lab team, as it has been difficult to purify proteins that contain GFP due to the size and the steric constraints. Back up, the team thought creating a construct that is fluorophoric tagged would be wise. The binding of this engineered chimeric flow body will also show two bands on the test strip confirming the presence of club root in a soil sample. Finally, our last case scenario would have a construct where we have inserted the team's engineered chimeric flow body into the backbone of a mouse antibody. The positives of this construct are that it is one that is most likely to bind to PBEL04 due to the backbone, although the negative is that this would then require an indirect ELISA test kit to confirm the presence of club root. This construct would possibly further the help of agricultural companies to target club root and confirm the importance of PBEL04 in the pathogen, as its protein presence in club root has only recently been discovered. This construct working would still widely be a success, although it would not be as convenient for farmers, which is our number one goal. However, the science would still be moving forward. Now let us show the initial successes of our engineered chimeric protein constructs and the protein-protein interactions it shows through computer molecular dynamic simulations. Prior to moving to the wet lab, we wanted to ensure that our engineered chimeric flow body was able to fold and bind to the desired epitopes on our protein targets. Our dry lab team accomplished this by conducting docking simulations using software called GRAM. The simulations for these models are still ongoing to ensure that the structure remains stable.
Speaking of PBEL04, we have designed another engineered chimeric flow body that we have specifically intended to target two domains of PBEL04, which have considerable overlap in residues. As seen from the MD docking simulation, we see our engineered chimeric flow body binding to the two desired domains containing our epitopes of interest. You can engineer chimeric flow body to allow for a detection system to be put in place and mitigation in the future. We designed multiple constructs so that we could optimize our protocols before bringing them to the wet lab. As you can see, all 10 chimeric proteins docked successfully, indicating that we have multiple approaches that may be viable in the wet lab.
What our team has engineered here is that we have added GFP to our chimeric flow body seen in the previous slide. Simply put, the protein sequence seen binding to PBEL04 has been modified to include GFP as a way of direct detection of protein-protein interactions between our engineered chimeric flow body to PBEL04. As seen from this molecular dynamic simulation, we see binding of our chimeric flow body to PBEL04. With the success of this simulation, this chimeric flow body will be a main construct the team will aim to work with. Furthermore, the team has also engineered various chimeric flow bodies. One, as shown here, is containing a terminal fluorophore tag, and another is inserted our engineered flow body into a mouse antibody backbone as potential constructs for use, which has seen similar docking success as a secondary way of detecting the protein-protein interaction. The team tried to be as thorough as possible in case the wet lab faced difficulties.
We have taken into account much of the feedback and suggestions we have received from judges, mentors, and researchers regarding the creation of our prototype. Through our mentor meetings with Christy, we learned that we need to communicate the progression and planning of our project more clearly in our presentations. Most importantly, we learned what modifications we would need to make to our prototype, which is why we are using an ELISA test as a stepping stone to prove that our design functions as we hoped it would. Our actual proposed prototype is that of a lateral flow test, but we would need to test an ELISA system first. Early on, our human practices team attended an agriculture expo where we were able to speak with various individuals who would be impacted by our project. We secured many valuable contacts there and plan to reach out to them in the near future. Furthermore, we learned more of the importance of the issue that we are attempting to address, and it was emphasized to us that our solution could provide significant benefits to the agriculture industry. Additionally, we were struggling to come up with a delivery mechanism for our mitigation system. So we interviewed Dr. Elizabeth Schultz and Dr. Dmitry Yevtushenko, who are both plant biologists. Dr. Schultz brought our attention to how our mitigation system could be taken up by the canola roots, while also focusing our attention on plant anatomy and how we could get our system into the plant cells. We researched several mechanisms and came across cell-penetrating peptides, which seemed promising, and discussed this idea with Dr. Yevtushenko, who referred us on to a researcher who works closely with this technology. We plan to reach out to him in the near future. Through further outreach, we secured mentorship and funding from Genome Alberta through our discussions with their Chief Scientific Officer, Dr. Hees van Rooijen. As much of the data yielded through computer simulations was rather promising, the next step is to test our constructs in the wet lab. This will take place over the course of the summer, and we will be performing the ELISA test that was mentioned above, in addition to other assays. We plan to continue to gather input from other experts and will implement their suggestions as our project progresses. With the results from our wet lab tests and the perspectives of professionals, we will present our final project at the iGEM Giant Jamboree in the